So today's video is going to focus on viral structure. And we're really going to be looking at the parts of a virus and the differences between viruses compared to cells. So let's first look at some major characteristics of living things. So one of the first characteristics of living things is that they need to be made up of cells. If you'll remember, cells are the smallest unit of living things. So anything smaller than that, like the organelles of the cell, they are not considered living. So they have to be made up of cells. They have to be able to do metabolism. They have to be able to utilize energy, right? So they have to be able to somehow harness the energy from food, whether that's they made the food or not, and they have to be able to use it. Essentially, they have to be able to do some form of cellular respiration so that they can get ATP. They also have to be able to maintain homeostasis. So when we're maintaining homeostasis, it's not just balance, right? So we're maintaining a constant internal environment. This is why cells have a cell membrane, so they can isolate their internal environment from their external environment, and they can maintain a constant internal environment, whether that's ranges from appropriate acid base levels or pH levels, appropriate temperatures, water, nutrient amounts, but they're able to maintain this constant internal environment. Cells, also living things, have to be able to reproduce, whether that's sexually or asexually, that doesn't matter. Right? So whether they're just doing, binary fission like a bacterial cell would do, or they're doing meiosis and producing gametes, like a mammal would. But they have to be able to reproduce on their own, and they have to contain genetic material in the form of DNA. So they can't just have any nucleic acid in there. They have to have DNA, that double-stranded in the order of those bases, determining their genetic information. So now let's look at a virus and determine whether that would fit those criteria. So first of all, we said that a virus had to be made of cells. A virus is not a cell. A living thing had to be made of cells, And a virus is not a cell. A virus does not have a cell membrane. It does not have, remember the four things that all types of cells had in common: prokaryotic, eukaryotic, it didn't matter, right? They had to have a cell membrane. They had to have cytoplasm. They had to have ribosomes, and they had to have DNA or genetic material, right? Viruses, no cell membrane, no cytoplasm, no ribosomes, no DNA. Viruses are also not able to reproduce without a host. So they need a host cell for reproduction, and we'll talk about that more in a little bit. They need they can't do metabolism on their own. They don't produce ATP. They don't do energy conversions. They cannot maintain homeostasis. So a virus is not considered a living organism. They also, or the fifth criteria is that they had to have DNA, but viruses could have RNA and not DNA. So viruses don't really check any of the boxes to be considered a living organism. So for as sick as a virus can make us, they're pretty simplistic in their structure and what they're made of. So a virus just has a few parts. So a virus is going to have to contain some form of genetic material, and it can contain that in the form of DNA or RNA. If you look at the two examples we have here, influenza, flu, and a bacteriophage. Let's go ahead and get this down. This is a virus that infects bacteria. Okay, so you'll notice this flu virus here has RNA for its genetic material, where this bacteriophage has DNA for its genetic material. So it has to have some form of genetic material. It has to have what we call a capsid. Okay, so this capsid here, this is almost like a protein shell. It's like a protein coat. But remember, we said that proteins are built by ribosomes. So this is kind of a ding, 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 red flag. Let's make a connection here, right? We said in our last slide that viruses don't have ribosomes. So somehow, to make more viruses, they're going to have to get some ribosomes somewhere so they can make proteins. And that's where that host cell is going to come into play and how a virus will make us sick. And we'll talk about that in the next video. We'll cover how viruses make us sick and how they reproduce. So the capsid is the protein coat that is going to surround it. Okay? Some viruses have an envelope. So this is only some viruses that have this envelope. Okay? That envelope is going to come from the cells they infected. That's actually part of the cell membrane from a cell that they've infected. And so essentially when the virus is actively replicating, it will burst out of the cell when the cell is all full of viruses, and it will take some of that cell membrane with them. Okay? That will actually make it easier for them to infect other cells because they now have this chunk of cell on them. And so it makes it a little bit easier for them to essentially trick the other cells into letting them in. So it makes it a little bit easier for them to infect other cells. Okay, and then some of your viruses will also have these protein spikes on them. Those protein spikes are going to help with attachment to cells that they're going to infect. Okay, and so you'll see those on some pictures and not on others. Okay, so that's about it for a virus. You don't need to memorize you don't need to know all the parts of a bacteriophage. Okay, but they're going to have genetic material. They're going to have this capsid that's a protein coat. Some will have an envelope. Some will have these protein spikes. So let's look a little bit closer at these protein spikes, or these are these surface markers. So these are the protein spikes. So the protein spikes that are on the viruses are cell specific. Let me explain what I mean a little bit here. So for instance, let's say you have a cold. The common cold is done by a virus. A common cold gives you different symptoms than the flu, and a flu is also done by a virus. So with a common cold, you usually have like a runny or stuffy nose, you have some congestion problems, maybe your throat's a little bit sore, maybe you have a little bit of a cough. With a flu, you usually have a fever and body aches. So what that should tell you is that those viruses are infecting different cells. The cold virus infects the cells along your nasal passages, maybe along your throat. The flu virus is affecting cells in different parts of your body. Maybe you get a stomach flu, a stomach virus, and so you're vomiting. You don't have a fever, you don't have congestion, you're just vomiting. Well, with a cold, you're not vomiting, which means that then your GI cells are not infected by the cold virus, but they're infected by the stomach virus. But the stomach virus didn't infect your nose cells or your throat cells. So the surface markers, or these protein spikes that you find on the viruses, they help determine which cells the virus is going to be able to infect. You'll see in this first image here, I've got receptor binding happening on this cell. So the cells will have certain receptors that allow that virus to bond to it for it to be able to enter the cell. And the virus has to be able to enter the cell for it to make you actually sick. And like I said, we'll talk more in detail about that on our other video. But the basics of what you have happening here, right, is our virus enters the cell. The virus uses the parts of the cell here to make new viruses. Then it releases more viruses out to infect other cells. So if the cell doesn't have the right receptor for the virus to bind to, the virus can't get into the cell. That's why we can have viruses that infect animals and they don't infect plants, or they infect plants and not animals, or viruses that infect dogs but not humans, right? Because of these different receptors and the different surface markers on the different types of viruses. Okay, so pretty straightforward. We need to make sure we're comfortable with characteristics of living things, the surface markers and cell specificity on the viruses and the parts of the virus. So as always, make your notes in your make your questions in your notes so that we can clear those up as soon as you come in class, so we can then do our activities and labs with this.